Stanford University.
I sort of fake it. And he also has expertise in aerobiology, so I asked him to talk to our class a little bit about aerobiology, because I think that that's one of the great untapped frontiers in astrobiology. And Rocco also has the distinction of being the only American PI, would we say, on some of these experiments that have gone up with the Europeans on satellites like Biopan and now the EXPOSE missions on space stations. So he's going to talk to you a little bit about that. And what these are is experimental ways to look at the concept of panspermia. So I think that the best thing is to just pass it over to Rocco, and then we'll finish hopefully a few minutes early, because I'd like to take a minute or two to talk about Thursday's class, because this is going to be wild and crazy, and you do not want to miss it. You want to be here bright-eyed, bushy-tailed, and ready to ask questions. So I'll pass it over to Rocco in the meantime.
Thank you. What I'd like to do is, I hadn't planned on doing this, but comment on that previous papers that look at weird life. And the first one was Paul Davies, who's he's a very, as Lynn said, he's a very bright guy, has won several awards. He is a theoretician. He's not a biologist. And one of the things I'd like to say about what was presented and the articles, if you should choose to go read the articles, and thinking about life, and that is one of the first things that we did with Viking was to look for life. And I don't know how many of you are familiar with the Viking mission. Viking mission took off in 75.
Everybody should be.
Everybody should be. Landed in 1976, two opposite ends of Mars, and with two identical landers. And it looked for, one of its main objectives was to look for life. Can anybody tell me what was considered by the life detection team and one of the overall scientists who helped plan it as the primary life detector, keeping in mind that we're talking about weird life here on Earth? Would anybody like to speculate on that?
Camera.
Exactly, the camera. Something walks in front of you, that's the first indication of life. And so one of the things that I would like to say with regard to how to think about, this gets back to what we're going to do later today with the Red Rain paper, is how to criticize and how to think about a process. Somebody's presented you with weird life, speculation on weird life, and that we can't see it because we're not looking for it. Well, the first thing you say, well, it's not big. If there's something crawling around that's big, I don't care if it is carbon-based or silicon-based. If a rock is crawling under its own power, you'd know that. And that certainly would be different life and different life based than we are. So we know that what he's talking about without him saying it is something that is small. And if we're talking about one of the things that was said in the article that I read is that when we look through a microscope, we look for life. We already have a preconceived notion of life. Well, true, we do have a preconceived notion of what life is. However, when you look under a microscope, if you are a microbiologist and you've spent a lot of time looking under a microscope and through a microscope, and I'm talking about somebody who's a microbiologist, microbial ecologist type, and not strictly a molecular biologist, when you look through it, you see all kinds of things. You see dirt, you see crystals. You have to be able to discern dirt and crystals from something that looks alive. And a lot of things that look alive aren't alive. They actually are some kind of crystal. A lot of crystals you might look at and you might think they're alive. But one of the things you do is if you look under a microscope and you take a slide and you see something crawling around, even if it's only a micron, you probably will think it's alive without knowing anything else about it. So I think you really have to think very critically about anything that you read, especially when it comes to something like weird life. We have looked for other kinds of life. I differ with Paul Davies in that we have looked for other kinds of life, and we haven't seen it. I'm not saying that it isn't there. I'm saying that I don't think that he has produced, in my opinion, a good, solid argument. The other thing, you alluded to another individual who gave a talk and looking at weird life in a test tube. Well, that person was Steve Benner. Steve Benner is a chemist, and what he does do is he looks at various different combinations of things in a test tube, chemical reactions that are self-sustaining chemical reactions that keep on going, and if you keep on going, meaning that they will continue and continue and continue for a while and then stop. one of the things that he said in this article was that, or he was asked, he posed himself the question when he said that we have some chemical reactions that we've put together in a test tube that could be synthetic life, because they will sort of perpetuate. And so he asked himself the question, is it alive? And he said, well, it cannot sustain itself alone. We have to have a graduate student standing guard over it and feeding it once in a while. And so he did qualify that. So as of yet, I would say that weird life, possibly, but quite remote. The other thing I'd like to say is that I think most people really will acknowledge the fact that on Earth, between the time that we have a record of life on Earth and the time that we know that life first, or that the planet first accreted and became the planet, that it wasn't all that long. And so if you look at it, you can say, well, if life originated on Earth, it's not a difficult process. And it wasn't all that hard to do. And I think also a lot of people will acknowledge the fact that there probably was more than one origin of life on planet Earth. And that's why you really do call, when you look at the tree of life, if you look at it critically and closely, especially to those people who really put it together in the first place, you will see that it is called the last universal common ancestor rather than the last common ancestor. Because what is thought to have happened is that life went through a bottleneck of some sort, and all life except one branch survived. I mean, all life except one branch was killed. Because there were many events that people think were sterilization events on early Earth, because it was a very traumatic and dramatic place, as I think other people will tell you in some of their other lectures. So that's just a brief introduction. You should always look at things very critically, look at them more in depth, before you just take somebody's word for anything, including mine about what I'm about to say. So please ask questions if something should arise. And today we're going to take sort of a virtual journey. And our journey will take us from Earth's surface, which we know is teeming with life. And then we will go to the troposphere, where we will look at some data that has been collected by balloons and aircraft. And then we will go above that to the stratosphere, and above the stratosphere to the mesosphere, where only sounding rockets really have taken any data, and then through the thermosphere and spacecraft, and then to the outermost bounds of our planet, the exosphere. And this is where the air is very thin, the atmosphere very thin. Atmosphere is very thin, and eventually the gases just dissipate out into space. And beyond that is outer space. And we're going to talk a little bit about our first step into it, panspermia, what it is, how it was thought of, originated in the past, and what we think now. And then we'll conclude with the red rain paper and the possibility of aliens invading our planet. Okay, so this is a lecture about life on Earth and how life on Earth may go beyond planet Earth and out there in a way. And then I'll generalize a little bit and talk about life beyond the home planet. Whoops. Oh. Sorry, it went backwards. Here we go. So the primary question we have is, what is the potential for survival, adaptation, and biological evolution, meaning Darwinian evolution, in the atmosphere and beyond? Beyond meaning outer space, for another planet. But first, we have to have that first step. And what is that first step? First step is to determine if there is terrestrial life that can survive. Not saying metabolize, grow, multiply, or anything, just a simple act of survival. And live away from Earth's surface. In the atmosphere, in the space environment, or on another planet, such as Mars. And why do we even care? Well, there are a lot of reasons to care, other than just being curious about it, or in addition to just being curious about it. There are a number of evolutionary questions to be addressed, such as the origin of life. How did life originate on Earth? What were the conditions on early Earth? And how did life survive? Because it was pretty harsh. It wasn't quite as pleasant as it is here today. It was actually quite harsh. There was a lot of UV radiation. It was a dramatic, active place. So it can tell us something about how life can sustain itself under harsh conditions. It can also tell us something about panspermia, and I'll get to the definition of panspermia in just a little bit. And it'll also help tell us, well, what is the probability of the fact that life originated on Earth, or maybe life originated somewhere else and came to Earth? And that is part of panspermia. Or as some people say, are we really Martians? We could be. You know, early in the history of the solar system, one of the things that happened was there was a lot of comets and meteors that were falling on the terrestrial planets, Venus, Earth, and Mars. And we know for a fact that there are bits and pieces of Mars on Earth. And probability has it that there are bits and pieces of Earth on Mars. So what's to say that life didn't originate on Mars and come flying through the outer space and landed on Earth, and that's where the first organism came? Or to say the same, the reverse. If we go to Mars and we find it, how do we know it didn't end up in a rock that was thrown from Earth to Mars? Because the Earth and Mars threw a lot of rocks back and forth in the early days, in their young years, believe it or not. The other big question is, what is the extent of the biosphere? We know that it does extend kilometers beneath the Earth's surface. And for those organisms that are beneath the Earth's surface, we know that they actually metabolize, divide, have a real life. They're not just transient. We do not know how high the biosphere extends into the atmosphere, and we do not know if the organisms that live in the atmosphere actually live there, or if they're just transient. Another reason is something called planetary protection. And planetary protection is exactly that. Should we protect other planets from ourselves, from our life and our trashing them? And do we need to protect Earth? Well, there are treaties out there that said that thou shalt not trash other planets, and we shall protect Earth and not try to trash it. So yes, we have to know if we bring an alien being back. And by alien being, I'm not necessarily talking about little green men. I'm talking about bacteria or prokaryotic type or simple single-cell type organisms. We should bring those back. Do we need to worry about them? And how do we protect ourselves from them if we do? Those kinds of things. So, what is panspermia? Well, as it was originally put forth in the late 1800s, early 1900s by Richter, Lord Kelvin, and Arrhenius, it was essentially that these living entities would travel throughout the universe and develop wherever and whenever the environment is favorable. Okay? From star system to star system, from galaxy to galaxy. However, there were a number of criticisms that were brought out about their idea of panspermia. It cannot be experimentally tested. It does not answer the question of the origin of life. It just sort of puts it off to somewhere else. And I may talk a little bit about this, but life really will not survive long exposure to the hostile environment of intergalactic space or intragalactic space. You just can't. It takes too long. And when you're out there, life doesn't metabolize. It can't fix itself. If it gets broken, it's broken. If it gets hit by radiation, the radiation accumulates. And in fact, even if you're buried within a meteorite and you say, well, I can survive in a rock and I have this house of rock around me, that's not good enough. Because over millions of years, the individual components and minerals within a rock, such as something as harmless as potassium, decays and goes through radioactive decay. When it does that, it gives off radiation. It gives off enough radiation that over millions of years, the organism will accumulate damage and chop up its DNA and die, not be able to recover. So that's one of the killers about even any kind of organism traveling long, great distances through the universe. It just, at least life as we know it, just can't do it because it can't repair itself. So what do? We now have sort of modified it. We talk about panspermia in a more practical sense, and that is the transport of life from one planet to another, because those distances and times are not that long, such that life could possibly survive in a rock. However, to do that, you need to travel from the surface of a planet through the atmosphere and into space, and then back on the surface of another body that is hospitable. So let's begin our little journey here, and we start with the troposphere. This is pretty elementary stuff, so I'm going to go through it fairly quickly. You have the troposphere. The troposphere extends from the surface of the Earth to about 7 kilometers at the poles and 17 kilometers at the equator. And believe it or not, it is in this bottommost layer that most of the atmosphere of the Earth exists. In fact, most of it exists within the first 5.5 kilometers. That's not that high, believe it or not. And I think we all realize that temperature decreases with altitude in the troposphere. And also, this is an important little fact to remember as we get and talk about the red rain paper at the end, and that in the troposphere we have air masses, and the air masses heat on the bottom, so they go up, hot air rises. As it rises, it cools. As it cools, it falls. So there is constant masses of air going up and down, causing vertical mixing in the troposphere. Within these masses of air, there are particles. So these particles also go up and down, and they're transported across the planet, also in the troposphere. There have been a number of viable microbes isolated of a variety of different kinds in the troposphere, especially the closer you get to the surface of the Earth, the more you get and the more biodiversity you get. So you have representatives of a number of different kinds of bacteria, archaea, and eukarya. And in fact, there have been a number of studies that have tracked all of these organisms, all these types of organisms, through space and time. In other words, are there seasonal variations? Well, yes, there are seasonal variations. And as you might expect, during the springtime and during times in which there's a lot of wind and things that raise things up, there are more organisms, viable organisms, in the atmosphere, in the troposphere, than otherwise. And you shouldn't be surprised to find that there's a lot of heterogeneity in what you find in the troposphere. And it's a direct reflection of what is right beneath it. So if you are over land, you find a lot more organisms that are associated with soil than you do if you're over the ocean. Makes sense. So, but the big question is, okay, we find them, they're alive, and the way that most of the studies have been conducted have been to go up there, collect air samples by a variety of different means, bring them down, and see if anything grows. Well, we know that we are unable to grow most organisms, so we're only capturing a small amount of the organisms that are up there. The other question that is still an open question is,
I'm just dusting.
Oh, okay. The other question that's an open question is, are these microbes, even in the troposphere, which is probably the most hospitable place in the atmosphere, are they just transient and just hanging out for a little while until they go back home to the surface of the Earth? Or can they actually live there? Do they metabolize and grow and divide? There have been a few studies that have been conducted that have tried to determine whether or not organisms can actually metabolize and divide while in the atmosphere. All of them have been shot down in some way or another. In other words, there is no study that has shown unequivocally that microbes or anything else can sustain itself in the atmosphere and divide and grow. So as we go up above the troposphere, we get into the stratosphere. and it extends from the troposphere to a height of 50 kilometers. And here the temperature increases with altitude, but the pressure is only 1,000th that it is at sea level. Sea level is 1,013 millibars, so here it's like 1.013 millibars. Here's where we find the ozone layer, and also here we've actually found some microbes. The density, of course, seems to be less, and it's been mostly spores and fungi that have been found, and that is reproductive spores of fungi, and spores of bacteria, particularly things of bacillus. A spore, in a bacterial sense, is a state of being in which the organism says, uh-oh, things are bad, I'm not really in a great place, I'm going to shut down. So they shut down metabolism, shut down everything, and try to dehydrate themselves and cover themselves with what's called a spore coat, which is almost like a armor. So it's protecting itself from radiation and desiccation and lack of food. So spores have been found and some fungi. And then above that is the mesosphere. Extends to the top of from the top of the stratosphere to 90 kilometers. Temperature decreases with increasing altitude. It's about a minus 100 degrees C. And this is the highest altitude from which viable microbes have been isolated. And again, spores, bacterial spores, and some fungi. And the highest was from a sounding rocket at 78 kilometers. That's it. Is that as high as there are microbes? I don't know. No one knows. Because that's just as high as we've been able to look so far. Because it gets very difficult when you get up there with the air so thin to actually collect something and see whether or not what you've collected is truly there or contaminated. And then we get to the thermosphere. And that's 90 kilometers to 600 to about 600 kilometers. Again, the temperature increases with altitude. It's about 1,500 degrees centigrade, and it's due to the absorption of solar radiation. But if you're a microbe or something else that's out there, it's not going to necessarily feel that hot. It's going to feel cold. And it's due to the low density of molecules. There isn't much gas up there. It's a vacuum, basically. And so what do you need to transfer heat? You need molecules and something to transfer the heat. So there isn't much of anything up there to transfer the heat. That's why it wouldn't necessarily feel cold. Just so you know, put it in perspective, the International Space Station orbits in the thermosphere between 320 and 380 kilometers. The ionosphere, part of the ionosphere is in the thermosphere and is created in the thermosphere, and it has not been sampled for microbes. However, microbes have been exposed and have shown to survive in the thermosphere. I didn't say multiply and divide, I just said survive. We'll get to some of those in a minute. Actually, you did hear in one of the earlier talks about the water bears, the tardigrades. Thermosphere is where they were exposed. And above that, and the last, and uppermost layer of the atmosphere is the exosphere. And it is through the exosphere that if you're going to go out into outer space, you have to travel and you have to reach escape velocity, and that happens to be 11.2 kilometers per second. So if you're going any slower than that, gravity will pull you back down, even if you're a microbe. And so one question I get asked is, what about microbes that, since we don't know, can they travel up all the way? Well, they might be able to travel up all the way to the exosphere, but are they going to go out into outer space on their own? Well, probably not, unless somebody gives them a real boost to go 11.2 kilometers per second. And it's up there, the air is pretty thin, and you get hydrogen, some helium, CO2, and atomic oxygen. And again, it is not concentric for microbes. So in summary, what do we have here about the atmosphere? I think we have a general idea of what the composition is. It's mostly nitrogen, some oxygen due to biological production, and then we have some trace gases, argon, carbon dioxide, neon, helium, and methane, which is mostly also biologically produced, krypton and hydrogen. And there's some water vapor up there too, which varies. The concentration varies with time and space. And so if we look at solar radiation spectrum at the top of the atmosphere, the red line, and at the bottom of the atmosphere, we see that actually now in present-day Earth, we're fairly protected from a lot of the UV radiation, which is down here. We're protected by the ozone layer, we're protected by molecular oxygen. Water vapor also is a big protective agent as well. So also in summary here, just to reiterate, that you have, as you go up in altitude, you have a decrease in temperature to a certain point, and then you have an increase in temperature, and then it remains stable. And then you have the atmosphere, of course, comes down. The density of the atmosphere keeps going down and down and down, until eventually dissipates out into outer space. Okay, and here we have the formation of ions in the thermosphere, and it also overlaps into the exosphere. So what's next is to go beyond Earth's bounds and out into space. And interplanetary space travel, and what are the ways in which you can do it? There are meteors or meteorites, and that's more what I call a natural way of organisms or things traveling from planet to planet. And this is actually regarded as lithopanspermia, in which you have a microbe or some life form embedded within a rock, within a meteor. Or spacecraft, because although we try to clean the spacecraft, and people always refer to sterilization of spacecraft before we go to Mars or someplace else, well, we don't really sterilize them. It's really bioload reduction. And that means that you reduce the amount of microbes that are on the surface to a known number that's actually quite low. I think it's like 3 Bacillus subtilis spores per cubic meter, which is pretty low. So let's talk a little bit about the history. Since we've been able to fly things, we have flown and exposed all kinds of things to the space environment, or as high as we could go. Viruses, vegetative bacteria, including Deinococcus radiodurans, which is noted as being very radiation resistant. Bacillus subtilis spores have been exposed, okay? And that was all during the 60s and early 70s, and at that time all died instantly except Bacillus spores. There was a classic experiment called Long Duration Exposure Facility Experiment, which was left up there from 1984 to 90. It was only supposed to be a year and a half, but they didn't have the money to bring it down, so they just let it go and finally got the funds to bring it down in 1990. So that's why it was up there for so long. And in that classic experiment, the only thing that really survived were spores of Bacillus subtilis, and only if they were in a multilayer or mixed with a substance such as glucose. If they were in a monolayer, they were essentially all killed within minutes by the UV radiation. Essentially all of them, all 100% survival exposed to the space vacuum, and 80% survival if they were in a multilayer. Multilayer meaning at least 2 or 3 layers, where the top layer sort of gave their all, they died. Those underneath survived because the top layers filtered out the UV radiation.
Rocco, could you explain what vegetative bacteria is?
Oh, excuse me, yeah. Bacteria, if you look at bacteria or archaea, or you look at prokaryotic cells, if they're dividing, metabolizing, and dividing, they're called vegetative. Anything in a vegetative state is alive and metabolizing. If they are in a spore state, they are essentially in a state of suspended animation. As I said earlier, what they do is they shut down. They shut themselves down. They don't reproduce. They dehydrate themselves. They cover themselves in a coat of armor called a spore coat. So they're non-metabolic, not active, not at all, non-vegetative. So that's the difference between vegetative and non-vegetative. One's active and one's not. One's actively metabolizing and living. The other one is not. It's sort of in a state of suspended animation. So, as you would travel out of the atmosphere and into space, why is it so difficult for life? Well, it is pretty bad out there. It's pretty hostile. There is cosmic radiation. There is space vacuum. there's solar wind, there's UV radiation, there are solar flares, solar particle events. And once you get out beyond even the magnetosphere, which extends actually beyond the exosphere, you are really exposed to all of these really bad things. Not to mention it can get to be pretty cold out there. So it's inhospitable to life and to metabolism. You can get blown away literally by the solar wind. You can get blown apart, or you can be ripped apart by solar flares and by cosmic radiation, literally. Literally. And so, okay, suppose you do that, you get out there and you've survived some travel through space, and you land on another planet, and it's not your home planet, it's foreign. So why might it be difficult? What are some of the reasons that may make it difficult for you? One is that there are differences in atmospheric pressure and composition. Here we have mostly 78% nitrogen, 21% oxygen. We like to breathe oxygen. Well, you go to someplace else like Mars, you don't have that. You don't have much air at all, much atmosphere at all. It's only 7 millibars compared to 1,000, and it's mostly carbon dioxide. So it's not really conducive to life like ours, or us. We also have an altered gravity regime. We're used to 1g. You go to Mars, it's less than 1g. You go to Jupiter, and it's 12g. There are a number of temperature differences. For example, you go to Mars, it's cold. It's really cold. It rarely gets above freezing, let alone the differences in the atmospheric pressure and composition. It's cold, very cold. Usually minus 70 or minus 80 degrees C, minus 90. That's the temperature, put in perspective, that's the temperature of solid carbon dioxide. And that's why you see on the poles of Mars carbon dioxide actually freezing out. And if you go somewhere, you have to eat. So what is there to eat? So there are nutrient source differences. You have to have, if you're like most organisms, you actually have to eat organic carbon. You can't make your own unless you're a plant or something related to a plant or an autotroph of some sort. You can't make your own organic carbon. So you have to have some kind of organic carbon to eat. And of course, you also have to have nitrogen. You have to have carbon and you have to have nitrogen in sufficient quantities to support life. And again, the atmosphere is very thin. It's only 7 to 10 millibars compared to 1,013 here. And the surface of Mars has some oxidants. So there aren't many things such as organic carbon right on the surface. So there isn't much in the way one might be able to find, or an organism might be able to find, even on the surface, for eating if you eat organic carbon. It gets oxidized away by the surface material of Mars. Now I'd like to talk a little bit, just briefly, a little bit about flying microorganisms in space that I have. I work on halophiles that are organisms that live in salt. They include organisms that live in the intertidal, okay, and these organisms that live in the salt crust of the intertidal, and this is a salt crust made up of calcium sulfate as well as sodium chloride and other kinds of salts, just the normal salts that are in the sea. And when it precipitates out, it traps the organisms. Most of those organisms die, but some of those organisms are alive and well. And you'll see here, this is a green band in this gypsum halite crust. And this green band is a cyanobacterium called Synechococcus. It gets its nitrogen from nitrogen fixation. It can take nitrogen out of the air. It gets its carbon from carbon fixation. It takes CO2 out of the air, makes organic carbon. And it gets its energy by photosynthesis in the sun.
Rocco, could I just ask you a quick
Yes.
question about the, you said that there wouldn't be any inorganic carbon on Mars.
On the surface of Mars.
It would be oxidized away by the
It is.
material.
There was an active experiment on Viking that did that, that showed that.
And what, and that wouldn't have been the same as the early Earth because they come from different bodies?
Okay, there is organic carbon. Organic carbon falls onto the surface of Mars today. We don't find any. We put organic carbon on the surface of Mars during Viking. It went away. It was transformed into CO2. There were a number of experiments that were then conducted in laboratories here on Earth to show that if you have an oxidizing material on the surface of the planet, it will oxidize the organic material and try to change it to CO2. So it's through those series of experiments and the results of the Viking experiment, where we actually put organic carbon on the surface of Mars, that makes us really believe that the surface of Mars is oxidizing. And we have not found any organic material there to date. Nothing. None of the Mars rovers, the Viking, nothing. Pathfinder, nothing. And we know that it does fall to the surface. Okay, I also work on organisms that live in solid sodium chloride. Okay? And this is a sodium chloride block, rock that we got out of a crystallizer pond in Guerrero Negro, Baja California. There's the biggest salt company in the world down there. And sometimes the salt precipitates out too quickly, traps all the organisms in it. Well, we were given one of these, and you can see it actually was pretty big. We put it in the van, drove it back. This is what it looks like. This is a solid sodium chloride crystal with the red. The red happens to be from beta-carotene that is produced by the organism. That's why it looks red. Beta-carotene is carotenoid, just like similar to the carotene you see in carrots. This is a photomicrograph of one particular organism, Halorubrum caoviatoris, and this is a salt crystal, and it's perfectly happy there. You take away all the salt, put it in distilled water, and it's not so happy. I throw this up here to let you know that the halophile salt-loving organisms are found throughout the tree of life, in the eukarya, the bacteria, and the archaea. And they're not just some side esoteric organism. They're actually an important part of evolutionary history, and they are easy, probably since they're found throughout the tree of life. They probably arose more than once, and it's probably very easy for them to arise in nature. I had the opportunity to propose an experiment to fly on the European Space Agency's Biopan facility, which went up into the thermosphere and exposed biological samples to the space environment. And I flew the Synechococcus that I got out of this gypsum halide crust, and I called it, at that time, I've got to update this slide, actually up until the last few months, we called it Haloarcula. And then we refined our experimental protocol, and it's now actually a Halorubrum. Not only us refined it, but they renamed the whole genus. Okay, so we flew it. I thought it might survive, not only because it has carotenoids that have the ability to protect against UV radiation, but these organisms are very, very well adapted towards desiccation. They can survive just being dry. So I figured if they can survive being dry, they have some protective mechanisms exposed to UV, that they might be able to survive exposure to the space environment. So we flew it. I asked the question, very simple question, not sophisticated, just can they survive? So we grew them, washed them, free of all organic material, put them on a quartz disc, put the quartz disc in the Biopan facility. We had two sets of discs, one that we exposed to the UV, they were on the top, another one that was beneath them in the dark, and we had another control set in the lab. Put them in the Biopan facility right here. This is what it looks like when it's open. This is what happens on the spacecraft. It actually opens up just like that and exposes the samples. When it's finished, it closes. It was put on the outside of a Russian rocket. Okay, the first one was quite a while ago that we launched the first one. The organisms were exposed for about 2 weeks. They got a heavy dose of UVC. That's UVB and C. These are the killers. UVA, we get a lot of that because it's not filtered out and it's the least energetic. UVB is more energetic and causes more damage. UVC is the most energetic and causes the most damage, and most people think that just about all of it is filtered out by the ozone layer, so we don't see much of that. Okay? So they got 10,000 kilojoules, and here, if you're on the surface of the Earth, you usually get about 6,000 kilojoules per meter squared. Okay, the satellite landed. That's what it looked like. Okay, opened it up. Samples were then sent back to me. We looked for survival by a number of means and looked at DNA damage. One of the ways we looked was looking at live dead stain, which is easy. If it's green, it's alive. If it's red, it's dead. And we counted the red ones and the green ones. And we did a variety of other things and then finally came up with some numbers for survival rate. And essentially, if you're in a vacuum, space vacuum, you get 100% survival. If you're in UV in vacuum, the Synechococcus survived. I believe that the Synechococcus survived because it was protected somewhat by some of the salts. The Haloarcula, or now Halorubrum, also survived. And the way we had prepared these is that they actually were in a monolayer, and they were not overlaying each other, and they were not most of them were not inside salt crystals. So these were the least protected. These, I think, were protected more by some of the salt crystals because the samples were harder to prepare. But nonetheless, the important thing here is that they survived. They weren't all dead. They survived. So my hypothesis basically was true, since it was simple. And I kept it simple because I didn't want to have to report a failure. Since it was simple, they survived. And this is the first time that non-spore formers were able to survive in a space environment. Prior to that, anything that was a non-spore former or a vegetative cell just died. Since that time, other things, as we have shown, could survive. And in Biopan 6, for example, a Rhizocarpum geographicum, which is a bipolar epilithic lichen colony, the whole colony was flown. And some members of the colony survived. And that's significant. The topmost members of the colony did not. They all died because of UV radiation. And as was pointed out earlier, there was another series of experiments, actually, that where they flew tardigrades, and you saw the results of that. The tardigrades survived, but they could not survive exposure to the UVC. UVC is pretty harmful.
Can you distinguish between UVC and gamma radiation? Were there filters that allowed you to know which radiation was?
Oh, these were not, none of these on Biopan were up there long enough to really get anything except UV radiation. When you're talking about any of the other kinds of radiation, you have to be up there longer for the probability of being exposed, actually getting hit by hard radiation. So, and I have another series of experiments that are up there now on the space station that are going to be up there for a year. And that's when hard radiation, ionizing radiation, gamma radiation, all of those things come into effect.
The woman who came from the European Space Agency last year,
Gerda Hornick.
yeah, Gerda, was pointing out how much the temperature goes up and down, too, as it revolves. Is that oscillation, possibly? Irregardless of what kind?
Okay, that's a really good question. But what happens when you're up there is that you're essentially in a vacuum, but you're in a spacecraft. Spacecraft is always spinning and tumbling. And there is an oscillation in temperature. As you face the sun, you heat up quickly. As you face away from the sun, you cool down quickly. In Biopan, the temperature oscillation was between, let's see, when you were up there, the temperature oscillation, it never got higher than about never got colder than minus 17 and never got hotter than 15 degrees C. Up in space. And there wasn't much in the way, there was no real thermal protection. It was just the way the things spun. If you're facing the sun, you're going to fry for a long, if you're up there for a long time and facing the sun, it gets hot. Because you can absorb the solar radiation. It's all absorption of the solar radiation. Does that answer your question? Okay, then I've also conducted a whole series of experiments where we looked at what could survive possibly on the surface of Mars, survival of spores in a monolayer under 12 microns of sterile sieve Mars analog soil exposed to increasing doses of UV radiation. basically, if you're under 12, even a 12 micron layer, which isn't very much, you can survive, or a spore can survive. You're desiccated on Mars, but you can survive. If you survive you can continue to survive in the atmosphere of Mars and Mars dust. So what we did is we took Bacillus subtilis spores again. We had a chamber, and we mimicked, believe it or not, a Mars windstorm. We put some Mars analog soil in the bottom, mixed it with spores, mimicked a windstorm with UV radiation pouring down on it, and then looked at survival of microbes, and sure enough, we saw microbes that could survive a wind-blown dust. Dust particles protect them. We actually knew that because we had that probably would happen, because we know that happens here on Earth. You get up in the atmosphere, you're a microbe, you're attached to a particle, you're protected much more than you are if you're a lone being out there. So in summary here, terrestrial life can survive away from Earth in a space environment and in a simulated Mars environment. And it helps address the origin of life and early evolution of life questions, such as survival strategies prior to complex DNA repair mechanisms. Because remember, when you're out there flying, you're not metabolizing, you're just there, accumulating all the damage and all the horrors that come upon you. You can't fix yourself until you get back into water. So there have to be some survival strategies prior to DNA repair mechanism evolution. And so I'd like to say that we've just taken that first step out there in understanding the process of what it takes to go through the atmosphere. Question? Yes?
When you simulated the windstorm, like how long was the simulation?
It wasn't, compared to a windstorm on Mars, it really wasn't that bad. It was, what did we go? I think we got the thing to go 3 weeks before it froze up.
And that was
It's usually, I have to say that in a lot of these simulation experiments, I mean, they're not exact simulations, they're close. But a lot of times the machinery just can't handle it. Microbes can seem to handle it better than the machinery. Yes?
Going back to the oscillation in temperature,
Mhm.
is that something that might also happen on an asteroid traveling between planets?
Yeah, but you're buried inside. In fact, that's a very good question, because if we recall, back in 1996, there was a science paper published by McKay et al. From Texas, Johnson Space Center, where they looked at a meteorite, ALH 84001. That was the first claim of, we think we have evidence of life in a meteorite from Mars. One of the critical questions that everybody asked then was, how hot did it get inside that meteorite? Well, it didn't get that hot, because actually the outside will will sort of buffer it from extreme temperature fluctuations on the inside. So there are a number of unanswered questions, such as, can microbes survive the meteorite ejection process? Probably yes. Can microbes survive the meteorite landing process? Probably yes. The reason why I say probably is because these are active areas of research, and the data that's coming out suggests yes. What are the most likely organisms that could survive? Well, we're still working on that. The more we test, the more we find might be able to do the job of survival. And what protection, chemical and physical, is given by the meteorite material during long-term exposure? There are experiments ongoing to do that. Most of them have had a variety of technical failures. One of the things that they've done is they've taken, you saw biopan that was stuck out there on the outside of a satellite? Well, they've actually taken rocks and glued them to the outside of satellites. Problem has been that most of those rocks fall off, and especially during re-entry. So they've had a variety of technical problems. So more experiments are required in space and in some good ground-based simulation facilities. All right. Now on to a discussion of this paper. And I present this paper, as I said last week, and it's been around for a while, not because I'm too lazy to look for another bad paper, but it's a classic one. It really is. And it is pertinent to particularly pertinent to panspermia and to biological systems. I mean, you can go out there and find lousy papers in every field, but this is a good one for this particular class. And And like I said, if anybody finds another good one, please let me know. So, okay, as you may have read about in this paper, there was an occurrence of, in Kerala, India, of red rain. It was rain. It's not uncommon. This is the monsoon season. One would expect it to rain from July to September in 2001. However, what was unusual about it is that it was red. Not every rainstorm that came down during this time period, by the way, was red, but some of them were red. In this particular paper, samples were collected and examined by microscopy, and there was an absorption spectrum that was measured. They also did EDAX and CH. This is X-ray diffraction, and they did carbon, hydrogen, nitrogen analyses. These are just elemental analyses. One of the critical things that they did, though, is they also looked at ethidium bromide, which is a chemical that people typically look at they look at nucleic acids using microscopy. If you put mixed DNA and nucleic acid with ethidium bromide, it intercalates. You know, it's a double helix. It intercalates in there, and it binds to the DNA. Okay, if you do it to a cell, the cell's now dead because it can't reproduce. Okay, because the ethidium bromide's in there. That's why you have to be very careful when you use this stuff in a lab. And then you put it under a microscope and you hit it with UV light, and then you look at it. It glows. Okay, it fluoresces. Okay. And it's a really easy, elementary test to look for nucleic acids. Okay, now, there are good things and there are bad things about this paper. So, now this paper, believe it or not, this paper went through peer review. Peer review is a process whereby scientists write a paper, they submit it to a journal, the journal then sends it out to so-called experts who are supposed to look at it and see whether or not it is fit to publish and to see whether or not it is logical and if they have any questions. Sometimes you'll look at a paper and you say, the data are solid. However, the author's interpretation of the data may be a little in question, and so you question that. And so you're always asked, are there any scientific strengths to these papers? What are they? So I'm going to ask you. You read the paper, or at least glanced at it, hopefully at least the abstract. And are there any scientific strengths to the paper? What's good about the paper? Would anybody like to volunteer and say what's good about the paper? Or what they think might be good about the paper? Yes, sir.
Well, a lot of times you run into problems with not having enough samples of things, and so at least they had like samples from lots of different areas, supposedly.
That is correct.
And lots of them, and it was observed by a lot of people.
That's very good. That is correct. You never base anything on one lousy sample, or even one good sample for that matter. Never do it on one sample. So that's correct. They did it through space and time. Anything else?
They seem to have several different types of tests to run on it, so at least they were looking at it from a few different
Exactly right. They did. They looked at it from different angles. It's one of the things you should always do. Anything else? I agree with you. Okay, I agree. And the other thing that they did, though, is the number of tests, the kinds of tests they seemed to try to do were appropriate. Ethidium bromide, I thought that was appropriate. Okay, now, are there any problems with the paper? I've listed a few that I highlighted here. After 4 years, there was no decay or discoloration. Claims of a cell wall or mucus layer, yet no protein or lipid analyses done or sugar analyses. Because keep in mind, what are they saying? They're saying that these are organisms, and that these organisms are aliens. They are not of this earth. So, if you're saying, I think I have an alien being here. First of all, if they think it's something biological and after 4 years you don't see any decay or discoloration, that to me would show some sort of problem of some sort. But maybe not. Okay, claims of a cell wall or mucus layer. Okay, if you think you have something that's a cell wall or a mucus layer, it seems to me, what are you going to do? You're going to see what it is. And if you think it's alien, you're going to see, okay, if it's not us, we have proteins. Does it have any protein? a simple analysis for proteins that anybody can do. Okay? They didn't do that. What else is a major component? Lipids. You can easily look for lipids. They didn't do lipid analyses appropriately. And they also didn't do any kind of sugar analyses. So if you think something is foreign, look for the things that might make it foreign. A lack of things that you would find commonly. They didn't do that. Are the elemental analyses data reasonable for an organic compound? Somewhat. Then we come to the ethidium bromide test. Okay, I explained to you how ethidium bromide works. So they should have shown a photomicrograph using fluorescence microscopy, and yet they didn't. They went to something that was, they considered more sophisticated, I considered it more complicated, and that is actually looking at it from another way using a spectrophotometer and fluorometer. Does anybody, before we move to this particular thing, anybody have any questions over that? Anybody have any comments?
I mean, could you explain more clearly what exactly they did with the ethidium bromide, according to the paper?
Well, I think this actually sort of explains it. I will. Actually, he explains it better here than is in the paper. What happened was, after this paper was published, a lot of the news media got hold of it. And they said, aliens are raining down. All right. Needless to say, that is sensational. And so a number of us got telephone calls and said, is this for real? And so we gave interviews. And one of the things that I was asked to do was to comment on it on a radio program. Well, this radio program happens to go internationally. And it just so happens that one of the listeners happened to be the senior author of this paper. I was very matter-of-fact in my presentation on the radio of the facts that I felt that they did not draw the correct conclusions. And I also commented on the next series of slides that I will show you as well. But this is an email that I received from, and I did not change one word, not one letter, no spelling, nothing. Okay, this is it. Okay, he wrote to me. He wanted to point out one or two things regarding our research on his research on red rain cells. According to you, we have not looked at the DNA under the microscope after ethidium bromide staining. The answer is, and it was unclear whether or not they really did, and I drew the conclusion that they did not. The answer is yes, we did it, but it is not glowing. And because it was not glowing, that's why they didn't show anything. It is not fluorescing under UV after being stained with ethidium bromide. That suggests to me they either didn't do it right, or they didn't have the DNA, or something was very wrong. So they did what they considered to be a more accurate method for DNA detection using fluorescent enhancement by using a fluorometer. Okay, and here he explains what they did. Okay, and so they did, and these data from the fluorometer actually are presented in the paper. Okay. We tested more than 10 biological samples as controls. That's good. They should have a lot of controls. And all samples show fluorescence enhancement except the red rain cells. Okay? So something was wrong with their ethidium bromide technique, clearly. Because no matter what they did, they didn't necessarily really see good fluorescence. Again, we have cultured this organism at very high temperature. We got some stunning results that this is an add-on here of research, continuing research. We are about to communicate these results. And so that, I've never seen anything more come out of it. And he's saying, personally, saying that these are definitely extraterrestrial microbes which are not known to us. That's not a great sentence. It needs more works to say scientifically. So what he's doing is, I understood this to mean that he's backtracking a little bit and saying, well, maybe they aren't. So,
Are you sure it's not a she?
yeah.
Oh.
Yeah. I'm not, yeah, this one. Okay. However, simultaneously, and not reported in this paper as remember, a scholarly paper should take into consideration everything that is known, all the work that has been done in the past and is currently going on, and some real investigation into what's going on. The Indian government, by the way, did not ignore the fact that the rain was red. So there really was red rain falling down. Okay, and so they went out and they collected samples. And in frames 1 and 2 here shows microscopic spores that colored the rain. Number 3, at 1 and 2, number 3 is rain samples with A, spores settled to the bottom of this bottle, B, rainwater evaporated, and C, what they call spores suspended in the rainwater. Okay? And 4 is biological. So I'm not surprised that it got through peer review. The Indian government, on the other hand, actually did the whole series of tests and published some things showing that the red rain really was from this particular kind of green algae. That is terrestrial, not extraterrestrial. So the extraterrestrial hypothesis rests on the assumption that a loud thunder and flash of lightning during a storm was an exploding meteor. A better causal explanation for a thunderous sound and flash of lightning is thunder, not an exploding meteor. So just the fact that the colored rains fell sporadically over 3 months, I think refutes a meteor burst event as the cause. Given that the spores grew in culture into a known species of algae, the extraterrestrial hypothesis seems to be without factual ground. And in fact, I'd like to recall a statement that was made during the press release of the ALH 84001 when very well-respected scientists, McKay et al. From Johnson Space Center, presented their data and saying that they felt that all the data taken together collectively, they felt that it was evidence of life on Mars. That there was an individual who was making a comment to that, and he said, well, this is an extraordinary finding, and extraordinary findings require extraordinary means to be proven. That was Jim Schaff, and Jim Schaff is a very famous, actually, what do you call him, a paleobiologist? Bill Schaff, paleobiologist. And he actually was quoting another famous scientist, Carl Sagan, who said the same thing. This is hardly what I call extraordinary means to, or anything extraordinary evidence to suggest that there is anything extraterrestrial. So, but there are a number of unanswered questions here with regard to this paper. How did so many local spores contaminate the rain, and why in only scattered areas during the 2001 monsoon? That's an interesting question. But I want you to think back of what I said happens in the troposphere. That it's dynamic, it's not static, it's not just air just milling around. It's air going up and down and up and down, and pockets of air, and they travel. They go up and down, but not just in one spot. They go up and down, they'll travel a little bit. So, there are unique rain patterns during 2001. They were highly unusual for that particular area during that year. A high rainfall weeks before, then low rainfall prior to and during colored rains may have led to an algae bloom and spore release. Spores picked up by wind and contaminated the rain before it fell from the clouds is probably what happened. And there was probably transport of local spores into local clouds followed by local rainout. Again, by this upward and downward movement of air masses in the lower part of the troposphere. And so it probably may relate to vertical mixing in the troposphere. One of the questions that arose, I'll tell you, well, I was going to ask, but I won't, because one of the questions that came to my mind as I was looking through this is, what is the possibility? Okay, we know that these algae were there locally, and we know that they were picked up by the wind, and they were transported into the lower part of the troposphere. But then they were there for a while. And there are lots of them, tons of them. And I kept thinking to myself, okay, since there are a number of questions here about what actually happened in the clouds, and they were alive and well when they came back down, and they were alive and obviously then alive and well when they were traveling in the clouds and staying up in the clouds, and the clouds were nice and wet. These are algae. These algae are photosynthetic, so they get their energy from the sun. These algae also have an awful lot of carotenoid pigments. The other thing that occurred to me is when I was looking through and reading all of the data in the papers, is that normally these algae look orange. And when it was regrown, they looked orangish rather than reddish. But when they were up in the clouds, they were really red. One of the things that organisms do when they respond to UV radiation is to make more pigments and molecules that will protect them from the UV radiation. And beta-carotene is one of them. And so the more beta-carotene that this organism makes, the more color it's going to have, and the redder that color is going to be. So what was going through my mind is I was wondering if this organism was metabolizing while it was up in the clouds and making more carotenoids. And if that's so, that'd be a fascinating thing, because that would be the first time that an organism might have been shown to do something like that, and it might help better explain some of the data. That's just a thought. Yes?
Are there other reported cases of rain containing spores of any other kind of algae, or even the same algae, just in different...
Not that I know of. I mean, rain contains organisms. You do get organisms coming down on the rain. But so many is what's unusual about this. That's what's unusual about this. Yes?
In the slide that you showed us about the Indian government and the test that they did, when they dissolved, so this is just a quick question, did they, when they, the spores were dissolved in the water, was that when they were able to grow into the actual organisms?
They actually, yeah, when they brought them down and they put them into growth medium.
So when the spores were in the clouds and if it was a relatively wet medium, why
It would be wet. And keep in mind that they never actually identified the species or the strain. So some species of organisms don't require much water around and are pretty efficient at gathering things up. So maybe they were slowly metabolizing and making more. But I don't know. That's just mere speculation on my part. I have no data to back that up. Any other questions? Now, the thing I want to get across is critical thinking, so that you look at a paper, you don't just take it for face value. You look at it and you say, were the data collected appropriately? Were these appropriate tests? Did they have sufficient number of replicates? Were those replicates reproducible? Were they enough like each other? What was the standard deviation on those replicates? When they say something is different or the same, is it really statistically significantly different or the same? Do they know what they're talking about? When you see something like that that says, okay, well, we did a lithium bromide test and we saw nothing, it suggests to me that they probably, and then they didn't do any of the other nucleic acid analyses. They didn't actually go in there and do a real analysis for nucleic acids. That probably something was not quite right. And the other thing is that they didn't try to grow them. If they did, they were clearly unsuccessful. They didn't show any really good micrographs. They really didn't do a thorough job of trying to understand what this was. And I don't necessarily fault them for everything, because they were not trained to do this. That's what happens when a lot of people go out of their field and don't completely understand it, and then don't go back to ask somebody or to try to redo things in a proper way. So I want you to learn to look at things very critically and not take them for face value. Yes?
You said that in the email that you sent, you thought that he was backtracking.
Yeah, a little bit. Towards the end, because he kept saying, well, maybe they're not definitively aliens.
Well, in the paper it says that they're not of terrestrial origin, but it kind of leaves open the question.
Well, that's right. But most people would think not of terrestrial origin, that it's extraterrestrial. And in fact, as I said, it raised a lot of attention, got a lot of attention from the news media, and everybody read that and said, oh, alien. They didn't just bother to say extraterrestrial or even qualify at all. They said, oh, aliens in the rain. So, yes, he left himself open a little bit there. You are correct. Yes.
As someone who's part of the scientific world, how much do you think that like time pressure is kind of what affected this paper? Like the need to kind of be the first one on the news about.
Well, I think a fair amount. It's sensational. I mean, if you were the first one to discover an alien or discover life on Mars, that is a big deal. I think so. Yes, there is a certain amount of
Do
pressure.
you see that in a lot of scientific papers that are published, especially when it comes to outer space and the presence of aliens?
I think most scientists within their field tend to be more conservative because they don't want to embarrass themselves. Any other questions?
Can you just connect it up more accurately with this whole aerobiology idea? Because the part that's not quite terrestrial, the fact that it was up there for months is maybe more dramatic than the amount that came down.
Well, actually, there are two things that I think are fascinating with regard to aerobiology, and one is that there was so much up there that came down. And the mechanism, and we think we understand some of the mechanisms for getting things up there. But two things we don't really know is how long are they up there? This gives us some idea. This is actually important because it gives us some idea of how long things may actually stay up there. And helps us better understand, I think, and nobody's really studied this, or I've never read the paper, helps us better understand the mechanism of how things come out. And that when you do get rain out, that it isn't 100% efficient the first time something rains down. Clearly, otherwise they wouldn't have had so many episodes of this. That there is another mechanism that allows these particles to stay up there. People are studying particle transport across great distances of the world, usually dust. What is not being studied along with that is the transport of microbes with them. The whole idea of aerobiology is to look at what organisms are out there, what can survive. And I have to say that if you were to really go back into the literature and look at some of the really well done, well thought out studies on aerobiology, they aren't always the most up to date. They're released slowly, but most of them have to do with biological warfare. One of the things that was done in the 50s and 60s and maybe even the early 70s is to look at what makes an organism survive in the atmosphere. What are the mechanisms that allow an organism to be transported? How do you know if you release an organism here, it's going to go over there? And what are the effects that it's going to have when it goes over there? What is the dilution effects? Can you engineer, some of the later studies were looking at, can you engineer organisms that are more worthy, being in the atmosphere and can survive in the atmosphere, in other words, desiccation resistance, UV radiation resistance. And so you look at those studies and you say, okay, well, there have been a lot of studies. They have done a lot of work. We now understand a little bit more about what allows microorganisms to stay aloft, but we still don't understand exactly what their behavior is or what their lifestyle is, if any, when they're in the atmosphere. We don't know really, and it comes down to asking the question, even with all those billions of dollars of research that were conducted, not just in the United States, the UK, other parts of Europe, Soviet Union, and China, we still don't understand exactly what it takes for an organism to survive, or if the atmosphere is really considered a part of the classic biosphere in which they can live and reproduce and metabolize. We still don't know that after all these years and all these studies, and it's still an open question.
Rocco?
Yeah.
When someone sees a sensational article like that in the newspaper or whatever, how would you go about being able to evaluate whether it's something that you should pay attention to or something you should ignore and throw away?
Well, does it make logical sense? If you were just to look at it from a logical sense perspective, does it appear to be logical? And if it does or doesn't, you should actually then try to get another perspective on it and go back and read some of the literature yourself. For example, going back to Paul Davies' AAAS talk and some of the things that were written about it. He mentioned arsenic in particular. And he talked about arsenic. And I am sure, in the article I read, I am sure that someone either asked a question or he was dimly aware of the fact that there have been a whole number of studies that were done on Mono Lake. Mono Lake is a lake in Northern California, not too far from here, that's full of arsenic. And what came out of that is that lake has a lot of organisms that use the arsenic. And what he qualified it, they use the arsenic for an energy source. In other words, they'll oxidize it, they'll reduce it, and as they transfer electrons in arsenic and change it from one state to another, they will harvest those, they'll transport those electrons through an electron transport chain, and they'll make ATP, cellular energy out of it. Okay. And a lot of organisms that live there do that, and they live very well, and they don't find arsenic particularly toxic at all. However, what he qualified that, and he said, well, yeah, we know that there are these organisms that live in Mono Lake, and he called out Mono Lake in particular, that live in Mono Lake, and they will utilize arsenic. He said, but they don't incorporate arsenic as part of their entire, and part of the cell. And he said, weird life might be able to incorporate arsenic. I think that's stretching it a little bit. I mean, look, when you look for things like that, that's stretching. I mean, arsenic is pretty toxic, yes, but not to these organisms. And a lot of heavy metals that we consider to be toxic are utilized by microbes as well. There are organisms actually that can live in radiation. I mean, if we can isolate, identify, characterize organisms that live in radiation, and when I say live in radiation, they'll live in radiation waste piles from nuclear reactors, then I think that we have a pretty good handle on looking for living things. And I always say, if you're looking for something, look for the obvious first. And don't necessarily think it's there unless you have some reason to believe that it's there. And I don't necessarily think that we have a lot of solid evidence to think that there is another shadow biosphere. That's my personal opinion. And I'd argue with Paul Davies even if he was standing right here. And he may see this. And I know I will see him again, because I saw him last week.
Well, I'm going to take, first of all, let's thank Rocco. A couple of years ago, I was asked to speak to a journalism class at Berkeley, and they asked that question. When you see someone present something, how do we know whether we should be giving it press coverage or not? And the first thing that I suggested, which Rocco hinted at, but I'm going to say bluntly, is look where the article's been published, and if it's been published.
Oh, yeah, I didn't say
if
that.
If it's something as extraordinary as an alien life, or anything that's really big news, there's a very good chance it'll be in Science or Nature, because they're rapid turnaround, high-impact journals. The second line would be something like Proceeds of the National Academy of Science. Those are very, very solid papers, just as articles in Cell and a bunch of other journals. They're very solid. They're not quite as rapid turnaround general audience ones, but those are going to be solid. So you look for journals that have a really good reputation.
For more, please visit us at stanford.edu.